Yo creo que hay muy pocas ocasiones en las cuales podamos tener a un científico del nivel del doctor Núñez en Fisabia dando una charla, así que muchas gracias. Luego agradeceros también a vosotros el que hayáis podido venir. Ya sé que muchos de vosotros venís de otros centros de investigación y habéis venido aquí pues adrede a escuchar al doctor Núñez, ¿no? O sea que muchas gracias a vosotros también. Antes de empezar la charla, me gustaría hacer una pequeña introducción, ¿no? Para que un poco aquellos que no conozcáis tanto al doctor Núñez, pues sepáis un poco el nivel de investigación que ha realizado. La mayor parte del trabajo que ha realizado ha sido en Estados Unidos, pero el doctor Núñez empezó aquí. De hecho, estudió Medicina en la Universidad en Sevilla. Y bueno, posteriormente ya se fue a Estados Unidos y realizó allí una estancia postdoctoral. Inicialmente estuvo en Dallas, en el Southwestern Medical Center, y posteriormente realizó una residencia de medicina que estuvo en San Luis, en la Universidad de Washington, y después hizo otro postdoc, otra estancia postdoctoral también en San Luis, y en ese caso fue en el Howard Medical Institute. A partir de aquí ya en el 91 comenzó, digamos, él como líder independiente en investigación, como associate professor, en la Universidad de Michigan, en el Departamento de Patología, que es básicamente donde ha estado pues hasta la fecha. En el 2001 le hicieron profesor, que sería lo que aquí llamamos catedrático. Bueno, pues un poco comentaros las contribuciones del doctor Núñez, ¿no? a la ciencia. Yo creo que su contribución a la ciencia ha sido extraordinaria. Comentar que ha publicado más de 400 artículos, pero no es solo, digamos, el número de artículos publicados, sino realmente el nivel, la calidad de los artículos. Para que os hagáis una idea del impacto que ha tenido, eso se puede básicamente observar según el número de veces que la gente cita tus trabajos. Bueno, pues al doctor Núñez lo han citado en más de 140.000 ocasiones, lo cual es algo, vamos, excepcional. Os voy a comentar brevemente algunos de los descubrimientos que ha realizado el doctor Núñez. Inicialmente, cuando él comenzó como associate professor, descubrió o estuvo trabajando con la apoptosis, que es un mecanismo de muerte celular muy importante, y descubrió unas proteínas dentro de la familia BCL2 que eran clave en la regulación de la apoptosis. Para que os hagáis una idea de la importancia, la apoptosis se ha visto después que está implicada en múltiples enfermedades, en cáncer, enfermedades cardiovasculares, enfermedades neurodegenerativas. Bueno, pues después comenzó a estudiar inmunología y descubrió dos receptores, el NOD1 y NOD2, que son los receptores, digamos, fundacionales de una familia de receptores que llaman los NOD-like receptors. Bueno, pues estos son uno de los receptores del sistema inmune más importantes que hay. Además, descubrió que uno de ellos, el NOD2, en una mutación del NOD2, eso estaba implicado o favorecía el desarrollo de una enfermedad, la enfermedad de Crohn, un tipo de enfermedad inflamatoria intestinal. No solo eso, sino que después descubrió más receptores NOD-like y identificó que algunos de ellos estaban implicados en la formación de inflamasomas, que son un complejo de proteínas que son muy relevantes para reconocer a patógenos y a moléculas del daño celular. Bueno, pues también los inflamasomas también están muy implicados en enfermedad. De hecho, se sabe, por ejemplo, que los inflamasomas están implicados en el desarrollo de la obesidad o de la diabetes. Y en los últimos años, pues el doctor Núñez ha estado trabajando en lo que sería en tratar de entender cómo el microbioma, ¿no? Ese conjunto de microorganismos que tenemos en nuestro cuerpo, pues regularía el sistema inmune y también nos protege frente a patógenos. Bueno, pues teniendo en cuenta los descubrimientos que ha realizado tan relevantes, pues la comunidad científica pues también ha querido reconocérselo y le ha dado múltiples premios. Y bueno, solo voy a indicar algunos de ellos. En el 2017 le dieron el premio al mérito por su investigación del National Institute of Health, del NIH de Estados Unidos. En el 2017 también le dieron el premio de la Sociedad Americana de Inmunología. 2 años después le dieron el premio de la Sociedad Americana de Patología y en el 2019, ese mismo año, le eligieron miembro de la National Academy of Medicine de Estados Unidos. Ya no me voy a alargar más voy a dar la palabra al doctor Núñez, pero yo creo que era importante que supierais un poco el nivel de investigación del doctor Gabriel Núñez. Así que muchas gracias. Y bueno, la charla será en inglés, pero si después alguien quiere preguntar en español o en inglés, como quiera, puede preguntar, ¿vale? Gracias. Y bueno, que disfrutéis.
¿Me oís? Voy a dar la charla en inglés, porque normalmente este tema me costaría un poco de trabajo, pero quizá lo pueda intentar en castellano la próxima vez. Solamente darle las gracias a Carles por invitarme. Quiero decir que yo iba a venir a Valencia en el 20, en marzo, y fue desgraciadamente la pandemia de coronavirus que me impidió venir aquí a Valencia. Entonces venía a España de nuevo y digamos que era una oportunidad de volver y dar el seminario. So what I'm going to talk to you today is about the microbiota host interactions. I'm going to focus on two different aspects. One of them is how do the microbiota protect us from pathogens, sort of beneficial effect. And also I'm going to talk to you in the last part of my talk to the other side, how some members of the microbiota can cause disease in humans or in animal models. So when we are born, we are sterile and acquire very quickly a number of members of the microbiota, which includes, of course, viruses, bacteria, fungi, parasites, archaea, and so on. And this occurs very early after birth. During the delivery, depends on the type of delivery, so the mother have a major effect of contributing to the first inoculation of these microbes, and but then, of course, during the first year of life and the second years of life, there are a number of factors which contribute to in the baby to have early microbiota populations that, with the cessation of the breastfeeding and introduction of solid foods, change dramatically. And I'm going to talk to you about the consequences of that later on in the talk. Now, all of us here in this room have a different microbiota. Even if you are a family member, you have brothers and sisters have a different microbiota. Perhaps it's because these early initial interactions are different. We are born in different hospitals. We are greeted by different nurses. Maybe your grandmother gives you the first kiss, which might be a different grandmother in other situations, or different nurses. So we are different, even brothers and sisters have a different experiences early on in life. And perhaps that could explain why even brothers and sisters have a different microbiota, although of course they are more similar than unrelated individuals. Now, the other thing is, and you probably know here because Carles is here, that is a tremendous interpersonal variation in the populations of the microbiota. This is at the bacterial level, and you can see two individuals here, and you see this is a major taxa populations are very different. Of course, this is perhaps very extreme situations, most people probably in between, but all the different cavities, like the oral cavity, or for example the vagina, or intestinal contents, they have on the skin different populations of bacteria. So it's a tremendous variation, the personal variation in the microbiota. Now, if you focus in the intestine, this is what the majority of the microbiota is located, and this is something to do with the ability of nutrients. We basically give them a site, a warm site, and also give them the food to survive. Well, that is the site with more food, in the oral cavity, of course, and now later on. So there's more food, is more density of bacterial populations are increased. It's just a matter of food, more bacteria. But the point of this particular slide is to tell you there are different bacteria located in different sites. You can see by the colors that the bacteria, for example, in the small intestine, in the ileum, is very different from the ones in the large intestine. This is something to do with also with the availability of nutrients. For example, here we have many amino acids and simple sugars like glucose. Well, those are radically diminished in the column. So there will not be these bacteria. Our metabolism is very different from the ones in the ileum. And these bacteria, for example, E. coli, Enterobacteriaceae, which lives in the ileum, cannot survive very well. Of course, also the oxygen concentration is very different. Here is truly anaerobic, so the only anaerobic, strictly anaerobic bacteria can survive, not here. So this basically reflects different metabolic requirements and different ability of nutrients in this site. Now, when the human, the animal dies, the microbiota also dies, and not just that, they eat you up as the last meal, basically. And that's basically the end of the microbiota. So it's a sort of long life interaction with our microbiota, and we have a very special relationship, and I will talk to you in a minute, which is often very beneficial. So what really drove these associations, particularly with bacteria, which we understand better? From the point of view of numbers, actually, there are more viruses than bacteria, but we know much little about the viruses that bacteria, but actually those viruses often the hosts are the bacteria themselves. So, but we know more about bacterial interaction for different reasons. So we think that a lot of the interactions drove this sort of symbiotic relationship, something to do with metabolic advantages. So you have a very diverse microbiota, basically help you to extract the maximum number of calories from the diet. And this is already flexible because we have a large community. So you change your diet, well, there will always be bacteria that can use those nutrients that will bloom there to give you the maximum extraction of calories. And of course, there are other advantages with vitamins, biosynthesis, and other factors that are very important. So most people will believe that these sort of metabolic benefits, mutual metabolic benefits, drove this evolution, and they sort of keep these bacteria in place. Now, one of the examples of that is the fact that hosts like us lack enzymes that can digest complex sugars that we eat, for example, from plants, from vegetables. So we don't have in the pancreas or any other site in certain enzymes that can break down those bonds between complex sugars. So basically, these bacteria, when they go to the colon, they basically specialize in basically attacking these sugars that arrive there, and they can basically benefit metabolically. They can use this energy source, carbon source and nitrogen source. But at the same time, they produce products that basically can be now taken up by the host. So this is one major driver of these interactions, that we certainly benefit the barrier, but also as an energy source. So what I've been telling you so far is that there are many things, good things, that the microbiota provide to us. I mentioned about metabolism and nutrient acquisition, and I mentioned complex carbohydrates. But also, of course, for a long time, we know the immune system is developed, particularly in the mucosal sites, through interactions with the microbiota. This provides many beneficial effects, not just only local, but also systemic. So in a way, we could think, for example, there could be sort of a normal priming or vaccination sometimes against members of the microbiota. Those antigens could sometimes have counterparts in pathogens. So in a way, you've been vaccinated by this microbiota because we can sense them and develop B cells, for example, or T cells. They are very important in protecting us from when the real pathogens come in. Now, the other main function that I'm going to discuss with you today, and that's how the catalyst also works, is colonization resistance. And this is basically the ability of members of the microbiota in different sites in the skin, but particularly in the intestine, to basically inhibit the colonization of pathogens that enter, in the case of intestinal microbiota, through the oral route. And this is a fundamental function that perhaps are provided tremendous protection to human populations and animal populations before the antibiotic era, which is basically most of our lives. You know, we've been around for probably 150,000 years, and antibiotics were introduced in the 1930s and 1940s for the first time, penicillin. So you look at that, the percentage is nothing. So basically most of humans are being evolved without antibiotics. So basically these guys have provided tremendous protection from this colonization. Now the other side, the dark side of this, is that the certain members of the microbiota can cause or contribute to disease. I will discuss in the last part of my talk inflammatory bowel disease to give you an example how this could happen. Periodontitis, there are some scientists here that work on that. We know that this is caused by poor hygiene, the hygiene in the oral cavity that drives the overgrowth of certain bacteria, which leads to the activation of osteoclast and the loss of bone around the teeth. So this is really caused by this bacteria, oral bacteria. Now, sepsis in our hospitals, I'm sure here in La Fe, here in Sevilla, working in the Bien del Rocío hospital, these patients come down with septicemia from bacteria that live in their own intestines. For example, often is E. coli septicemia, or Klebsiella pneumonia, for example, lives in the ileum. These bacteria, they don't come from the doctors, from the nurses, or the environment, come from the own patient themselves. So members of the microbiota can kill the patient. This is one of the most common causes of death in human populations. There are many other associations, and the list goes on and on, probably hundreds of associations. I don't think that we have a clear understanding, in human populations, how relevant is these interactions. These are basically causing the disease. These are maybe perhaps indirectly contributing to the disease, but we really don't know much how important the role is. So going back to colonization resistance, so these members of the microbiota, these symbiotes, form a very stable community that resists change. So for example, if you go out and buy yogurt with live microorganisms in the supermarket, in the corner here, those have live microbes, but we know that they don't last very long. Basically, our microbiota takes care of them very quickly, so you cannot detect them for a long time. So the probiotics, they don't last very long when you're introducing to a person with an intact microbiota. And so it basically prevents the invasion of probiotics, like I mentioned, from yogurt, but also, of course, pathogens themselves, going to be discussing with you today, but also expansion by pathobions. This as a term which is being often used to describe members of the microbiota. They can cause disease. So they are symbiotes, but they can cause disease in certain situations. So this is one of the early papers in 1954. This is from the University of Chicago, which introduced these contents of protection of pathogens from the oral route by the microbiota. So in this case, these investigators were studying oral infection by Salmonella. And so here you have a group of mice that were treated with streptomycin to deplete the microbiota, or they were left untreated. And then these investigators infected group of mice with different number of bacteria from less than 1 to 10 million bacteria. Now, the ones that are controlled, that they basically were just infecting them, you need around 1 million bacteria to infect 50% of the mice. These are 24 hours. If you do the same experiment with mice who were treated for 24 hours with streptomycin, you need 1 to 10 bacteria. So the effect is about 100,000, and sometimes in some cases it could be 1 million fold. I'll show you in a minute. So this is a tremendous protection they confer, which probably in the area before antibiotics was very, very essential for the survival of individuals. When the water was not, water was not clean. There were no city halls cleaning the water or anything like that. So there was a lot of bacteria around, and these many humans would require a very high dose of Salmonella, for example, to cause infection, because most cases they would not even know. the microbiota will take care of the bacteria, and they will be totally unnoticed. So we did some experiment. This is actually unpublished, but to see how long, when this occur. So in this case, we treated mice with control with antibiotics, and we give them an infection. It can be done with Salmonella, or in this case with Citrobacter rodention, which is a pathogenic E. coli, which is a natural mouse pathogen. So we look at 12 hours here, and you can see, we look at different sites, in the ileum, in the lumen, in the tissue of the ileum, in the cecum, in the column. You see, and this is 12 hours. This is total bacteria. This is per gram, so basically the same, with different how you express it, total bacteria or just by gram of tissue. So you can see that even at 12 hours after infection, the difference here is about 1 millionfold in the ileum, and this is the same thing here with the column, the lumen. You know, this difference. Basically, you hardly can colonize. This is almost the level of detection is here, of detection. Well, compared when you give them antibiotics, you can see this is a log number. So this will be talking about 3 to 6 logs difference. With this experiment, 6 hours, still you have 3 logs difference. This occurred very, very early event. So how this work? So many people have been trying to understand the mechanisms how this occurs. So there have been 2 basic mechanisms that people have proposed, and there are data to support both. One of them is that this is through direct interactions that the microbiota, members of the microbiota, can counter the pathogen directly through a direct mechanism by different mechanisms that's being described. This includes competition for certain nutrients. Also, some members of the microbiota can make molecules. They can kill particular pathogens. So this is the direct mechanism. Others I can discuss with you later on. The other mechanism that is being suggested is that this is through indirect mechanism. Basically, the microbiota can stimulate locally host immunity, and then they can produce molecules like antimicrobial peptides. They can directly attack the pathogen. Now, none of these mechanisms seem to be specific for a pathogen, because it's very difficult to imagine you could discriminate specifically. There could be groups of bacteria, but not specific for a given pathogen. The other thing is that it's very likely that different pathogens may have different mechanisms. They will be part direct or indirect. The idea I want to tell you, when you're looking at 24 hours, the classical definition of colonization resistance, which occurs within 24 hours, and I show you occurs even by 4 hours, we can see this, 6 hours. We think that this is mostly direct interactions through the microbiota countering the pathogen. If you look later on, a week later, there will be more host-derived mechanisms, but the main mechanism of 100,000-fold was described in Chicago in 1954. That is seemed to be very too early to say this is something to do with immunity. Most likely it's indirect interactions. I'm going to show you our data on that regard. So we have been using different models of pathogens. This is the one we like a lot. It's Citrobacter rodention. It's like enteropathic E. coli, EPEC. We like it because this is a natural mouse pathogen. So the host for this pathogen is basically the mouse. So it's very physiological. And it's very, when you sequence the genomes of Citrobacter rodention, it's very similar to EPEC. All the virulence factors are there. About 80% of the genes are there. It's really, and the pathology is very similar. We like this model a lot to work in the mouse. If you infect with a human strain a mouse, there's no colonization, basically nothing. So this pathogen is being adapted to replicate in the presence of the microbiota of the mouse. So this colonizes the mouse, and inflammation is restricted to the gut. So it's very different from Salmonella. They can spread, go to the liver, go to the spleen systemically. This is basically an E. coli with virulence factors on it. So they like to leave close inflammation in the intestine. And often, particularly in adult humans or mice, this is eradicated by 3 weeks. Basically, at the end, you clear the infection, you get the inflammation, the diarrhea, but at the end, it's all clear. So they can get rid of the pathogen. So we started this work about 15 years ago. This is our first paper on this topic. So when you infect conventional mice, what I mean by that is that these mice which have microbiota, these are the regular mice that we use from commercial sources, basically the pathogen goes exponentially, and then by 21, 23 is eradicated. It plateaus at day 7, between day 7 and day 10. If you do these experiments in germ-free mice, what happens is they go 1 log higher, so 10 times more, presumably it's more space for them to grow, and then it cannot be eliminated up to 42 days. Now, what is remarkable about this is that now the entire microbiota of the germ-free mouse is basically a pathogen, but these mice, they have diarrhea and so forth, but after a while they look totally normal. They could look around, it's like nothing. So we were at that time, we were very surprised and puzzled about how a mouse could survive with entire hundreds of trillions of pathogens living inside, and it was like normal. And I will show you why was that in a minute. So we said that microbiota is required for the elimination of Cytobacter rodention. Now, there was no early alterations. The reason that there is not a clear infection is not because the lack of microbiota impair immune responses. Everything we measure, there was more antibodies in the T cells, antimicrobial peptide, everything was normal. So that was not the problem here, why they cannot clear. So we have to focus on the virulence factors that this pathogen uses to replicate in a mouse in the presence of the microbiota. So it have this locus, it's called LEE, locus of enterocyte effacement, have 5 operons and 42 genes. Most of the genes control activities like a type 3 secretion system, which basically inject effector proteins, a number of effector proteins encoded here, into the epithelial cell. So it binds very tightly to the epithelial surface. All these genes have receptors and a ligand to bind to the epithelial surface, which is called intimin and TER. So basically, they are included all here. So this is essential because if you knock out any of these genes, it's basically impair of the colonization of this particular pathogen. All these factors here in this island are controlled by a global regulator. This is a transitional factor. So basically have sites in the promoter regions of these operons. So when you turn LER, all of them are turned on. So it's turned on. So to look when this virulence factor was expressed, we made a strain of Cytobacter rodention, a reported strain, in which the lead promoter was linked to a chemiluminescent cassette called LUX. So by doing that, you put in germ-free mice, you put now the pathogen, now when it's turned on, LER and all the virulence factors become red. So you can see in the second express the virulence, it also is in the ileum, the last distal part of the small intestine, and also here in the rectum. Now, at day 14, when it's already past the inflammation, you can see that is already down-regulated the virulence. Now, this is not strict, this has nothing to do with the lack of pathogen because this is germ-free mouse. So the pathogen load is the same. The pathogen show you in the curve before. So it's nothing to do. I will show you later on that this is actually reflects selection of the pathogen. There are some virulent and avirulent pathogens. Some express LER or they don't express LER. So this, I will show you later on that basically what's happening is that the LER positive strains are eliminated while the LER minus are retained. And that's why it looks downregulated, although the bacteria is still there. So to understand how these virulence factors are used by this pathogen, you have to understand where the microbiota is located in the small and the large intestine. So this is the single epithelial lining here, single line of epithelial cells. At the crypts here, you have stem cells and Paneth cells. The Paneth cells are loaded with antimicrobial peptides, which are released when they sense the microbiota. You can see the mucus layer here in yellow. Typically, they don't have microbiota here because these antimicrobial peptides released from the Paneth cells kill the bacteria. So this is basically sterile. The bacteria are here in the lumen. Now, in the colon, it's slightly more complicated because you have 2 layers of mucus. The one layer of inner mucus, very close to the epithelial surface, is very dense. so the bacteria cannot go across. So this is very thick, and there's no bacteria. In the outer mucus layer, there's actually a loose mucus, very rich in mucus. Of course, the mucus comes from the goblet cells. They are glycoproteins. The bacteria specialize in living here because they can take advantage of this food, which is basically rich in sugars and in amino acids. So basically, they live here. But basically, near the epithelium is no bacteria. What I'm going to tell you in a minute is that these layer virulent factors allow the pathogen to go and attach to the epithelium, and that way they can use these virulent factors to replicate, acquire nutrients, avoiding the competition from the microbiota. So to do this experiment, we got germ-free mice, we infecting with wild-type bacterium or the mutant, virulent layer mutant, and we look at day 5 when its virulence is on, and at day 21, when we saw this downregulated, I look by electron microscopy. And you can see that most of the bacteria at day 5 in the wild-type bacterium is attached to the epithelial surface or very near the epithelium. At day 21, these are germ-free mice. The bacteria is still here. It's just in the lumen because they lack the virulent factors. They cannot attach to the epithelial surface through factors that are controlled by layer. This is at day 5 by rodention. It cannot localize at all because they lack the factors that allow them to attach to the epithelial surface. So the bacteria actually, if you look at by fish staining here, you can see the signal is basically day 5 is near the epithelial surface here. You can see this is yellow is a probe for rodention. At day 21, they're in the lumen. You can see, okay, but they're still there. And the mutant layer cannot localize either 5 or the 21 in the only in the lumen, cannot attach to the epithelial surface. So then we ask, okay, now the pathogen is attached to the epithelium. Now let's see what happens if we transfer the microbiota to the germ-free mouse, which have the rodention in the epithelial surface. There's some competition, but minimum, only like 50-fold competition. But if you do this experiment at the 21, where the pathogen is in the lumen, see what happens. You basically, it's 100,000, 1 million-fold competition now. So the layer controls the ability of limit the competition from the microbiota. Compare this curve with this curve. You do this with the mutant layer, you see it's the same competition because now it's in the lumen. So basically, all the data suggests that this strategy of using layer virulent factors to attach to the epithelial surface and avoid the competition from the microbiota. Okay, and that is basically location is essential. You are now where the microbiota is located, they overwhelm you, so you don't want to be there. You go to a site which basically is no competition, and they get nutrients through the epithelial surface. Well, if this is true, you could ask this following question. So this is in conventional mice. The wild-type bacterium grows, I told you, and then by 21, 23, I eliminated. Now, this is the mutant layer. This is with conventional mice, SPF mice. They cannot colonize at all. Here, they cannot go to the lumen. They cannot have no chance to go no competition. But look now in germ-free mice. It's no difference. Now, the layer virulence is not required. Why is that? Well, these layer factors is required for replication of the pathogen in the presence of the microbiota. So you don't have microbiota, you don't need the virulence. Now, why is that? Well, they can get the food from the diet. There's no competition. The mouse are eating, okay, you have microbiota, you have now to deal with the microbiota, they're eating the same nutrients you need to grow. Well, there's no microbiota, there's no competition. All the food they're taking is so you don't need the virulence now. You don't need to go to the epithelium. They just eat in the lumen, it's fine. You just get all the nutrients. At the end of the day, what they want is the food to eat, to replicate, and to pass the progeny to the next host, okay? So I don't have time to tell you the immunity part of this because by publishing a number of papers we published over the years. But basically what we found is that at the beginning, during the infection, there are two populations of the pathogen. One is called layer positive and layer minus. It spreads high levels of layer or very low levels. And this is early in infection. the layer positive are the one that can bind to the epithelial surface. Now, this is sensed by the host already as a danger, so now produce cytokines like IL-22 could reinforce the barrier. So this forces a stress situation to the epithelial surface. If you have mice which are deficient in IL-22, then the animals die by day 12. These major ulcers because they cannot maintain the barrier. So the IL-22 knockout, they're dead by day 12, all of them by day 12. Now, by then already, the adaptive immunity is induced and also innate immunity. We found that the B cells, the plasma cells, produce antibodies, IgG. They can bind against virulence factors controlled by LAIR, like TIR and intimin. So basically, they opsonize, we use that term to say decorate, put on the surface. And now neutrophils come in and engulf the LAIR positive. So basically what happened is, the only thing that remains in the LAIR minus, that's where these mice, the virulence down-regulated, and also the mouse is totally normal, because it's like an E. coli. Now, it's an E. coli. Cannot bind here, that produces the damage. But they look like E. coli. That's what these mice at the beginning were basically surprised. I could survive. Well, they are LAIR minus. Now, if you take this now and infect a new mouse, they turn on LAIR again, and they produce another cycle. So it's not like a permanent mutation or anything like that. This is part of the cycle. Now, in normal mice with microbiota, these are taken out by the microbiota. So at the end, you basically clear totally the pathogen. So it's good for both. For the animal that clear the pathogen, and for the pathogen, it basically replicate and pass to the new host if it's needed. But basically at the end, it's a good shake hands together, both survive, except if you're a child. With EPEC or mice, you infect animals, we'll show you in a minute, they are very young, they die. And I'll show you why they die in a minute, okay? So, as you know, newborns and children less than 1 year old are very susceptible to enteric infection. Any pediatrician knows that. Not just to enteric infections, many types of infection, newborns are very susceptible to infection. And the list, it goes here, including also EPEC, which is here, but also Salmonella, but many types of infections, viruses as well, okay? Everybody knows that. Now, traditionally, people thought that this is due to the fact that the immune system of the newborn is immature. And this is true, but typically happen within the first 3 months. After that, in newborns can make good antibodies because we vaccinate children when they are 5 and 6 months. That's why we vaccinate them. And they make good antibodies, IgG. So yeah, it could happen during the first 6 months, but after that, and they are susceptible to infection. So why that? Well, we reason that perhaps the microbiota could play a major role, not just the immaturity of the immune system. So this work was done by Yoongi Kim and Kei Sakamoto. Both of them are professors in Japan. So consistent with this phenotype, you get neonatal mice, 4 to 7 days old, and this is the adult, 7 week. You infect them, for example, with Salmonella. Basically, the adults all survive, but 80% of the neonates die. And you can normalize this by weight, but everything, they die. I'm a pathologist, but you don't have to be a pathologist. On the top, you see the normal epithelium with the epithelial surface. Here, it's all necrotic, all the epithelial surface, okay? Now, you do this with Citrobacter, it's even more dramatic. This is 100, this is 0. All of them die, okay? All of them. So how do you know if this is the immune system, the age difference or the maturity, or is the microbiota? Well, that's a complicated experiment. So we reason that we basically could have fixed age. We use adult gen-free mice, so the same age. Now, we transfer cereal contents from different microbiotas at day 4, at day 12, 16, 17. We keep them in an isolator. We just give them the regular food. If you look a month later, this is the microbiota from day 4. They come from day 4 mouse, in adult mouse. This blue line, these are basically lactobacilli. So this is lactobacilli because they used to get milk from the mother. So they still retain that population from the mother. You can see how different. Now, then the microbiota from the 16 just look already like an adult. This coincides with the introduction of solid foods in the animal. And when the mother, basically, some of the animals, they are breastfeeding, but also they can eat some solid food. So they become, this is like an adult. So now we can do the experiments. The only difference is the microbiota. The age is the same. So that's why you do the experiment. Otherwise you cannot conclude anything. So you do this with 4 days. You see here, the 50% of the animals die here. So we transfer 7 weeks for the 8. This is the adult mice, germ-free, put salmonella. You can see the pathology is very clear. You can see the neonate, a lot of mucosa edema, a lot of necrosis, a lot of neutrophils. You can see here. This is the adult, totally normal looking. The pathology score day and night here like this. You can do this the same with this is so when this when this sort of protection occurs, or lack of protection. This is germ-free mice here. You can see the microbiota from a day 4 or day 12. They don't have colonization resistance to Citrobacter rodention. Basically, they don't bring colonization down compared with this. So this is about 100,000 fold. This is exactly what the original paper in 1854. Okay, and then at day 16 you can detect some colonization resistance. Citrobacter rodention is even more dramatic. It's about 1 million fold, 6 log difference. The N4 and N12, they don't have colonization resistance, shown here. Okay, so what is the difference between an N4, N12, and A16? Well, the majority, the populations are very different, like I showed you before. The main difference is that humans, neonates, or mice with their breastfeeding, mostly have facultative anaerobes. So basically, they are Enterobacteriaceae, lactobacilli, but they lack obligate anaerobes, like Clostridiales and Bacteroidales. So we reason that perhaps those are the ones which are missing in the neonates, which are doing the job of blocking these pathogens. That's basically reasonable. But how you prove that? So what we did is that we prepare consortia of Bacteroidales and Clostridiales from our mouse colonies, and then we get a 14-day-old with lack colonization resistant, and we added them here to see which one they use protection. And the data was very clear. Only Clostridiales could be able to bring down the colonization. They can protect from pathogens, from this case from Salmonella. You can see here, for example, this is with N4. You see swelling intestine. You don't see the fecal pellets because it's all water, because the edema. You see the cecum is shrunken here. Look at with Clostridiales. They look very nice looking with the pellets. The pathology, this is like this. This is like this. You can see the difference. You don't have to be a pathologist. The Clostridiales are the ones that are protecting you from Salmonella and Citrobacter infection. This is basically another way. I'll tell you this. This is a case with rodention. You can see you can add Clostridiales, and you can bring now the colonization resistant, but the Bacteroidales cannot. Okay. This is more physiological in the sense that these are neonate mice, like 8 to 10 days old. We give them Clostridiales the day before, and then we inject them with Salmonella, and you can protect 50% of them. Okay, this is a normal mouse, a neonate mouse. So the conclusions is that the lack of colonization resistance was caused by the absence of Clostridiales in the neonatal microbiota. When you give them Clostridia, then you restore colonization resistance. And there's some work now coming from all the investigators who support this concentration, this sort of conclusion. I would not say that the Clostridiales provide all the colonization resistance. This is a very complex group of taxa with many different bacteria, and also it could be contribution from other bacteria as well. But at least the bulk in these models are Clostridiales. Has been shown also by other investigators. Now, importantly, this was independent of host immunity, because mice which are deficient in NYD8, TRIF, adaptive immunity of IL-22, still Clostridiales was able to protect. So look like this independent at least of these factors. It appears to be more direct interactions between Clostridiales and other bacteria and the pathogen. So to do the mechanism, Gustavo Caballero Flores is a postdoc from Mexico, came to the laboratory. He's a geneticist, he's a microbiologist. So he decided to attack this problem from a genetic point of view. And this work was actually published about 2 years ago. So what he did, Gustavo gets Citrobacter rodention. He knocked out singly all the genes in Citrobacter rodention by troposon insertion. So he knocked out 90% of the genes one by one. So we have a colony, a library of mutants with a single mutations in basically 90% all the genes in Citrobacter rodention, okay? And the 10% that remain is because they cannot grow in culture, so we could not study them. They require for growth in medium, so we cannot expand the colony. So the idea is the following, is to infect this library of mutants into gene-free and mice with microbiota. So if you find genes which are required to grow in SPF with microbiota but not in gene-free, those will be the genes which are used by the pathogen to evade the microbiota, because you don't need them when it's no microbiota, means they're not required. So basically the genes are required to grow in the presence of microbiota but not in gene-free mice, which include the LER factors I'll show you. And that's one of the factors that we found here, see? So we now selected we infected this, we collected the feces at day 2 after infection, we plated with Illumina sequencing, mass sequencing to quantitate the abundance of each mutant, mutant. And when you do, we selected mutants that they were basically, they cannot grow in SPF mice, but they can grow in gene-free mice. These are these, SPF minus GFF here. So this is minus, so they cannot grow. About at least 100-fold difference, so major phenotypes. What are these genes? If you look at here, there were 4 main groups, but the one that caught our attention is amino acids. There were this group of, each of these is a gene, operon. These are required for amino acid biosynthesis. So the pathogen can make their own amino acids. And this, if you knock out any of these, you cannot colonize in the presence of the microbiota, but gene-free mice, as this is shown here. So this is in gene-free, in blue. You can see this is a 5 amino acid here. These are the operons required for the synthesis of each amino acid. You can see that they can grow in gene-free mice in blue, but they're very highly deficient in red when this microbiota. There were also nucleoside metabolism and LPS, which is a cell wall component. So this is then we engineer the mutants again to make sure there's nothing to do with the library itself. So this is the first experiment. We make a single mutant of the 5 different amino acids. They can grow when you give them a medium-rich amino acid. All the mutants grow like the wild type. Now, if you use minimal medium with no amino acids, they don't grow. But if you supply them each amino acid that you make mutation, you can rescue them here. So this is the mutation by each amino acid they can grow. And this is very specific. So it tells us that we have made good mutants. What happened in vivo? So we infected the wild type and mutant 50-50 into a mouse. And you can see that in SPF mouse, the wild type can grow well, but the mutant deficient in amino acid biosynthesis, in this case arginine or threonine or histidine, they cannot grow. Here, see that. This is the gene-free mice, they don't have much an effect. What we notice also is that these genes during the infection were induced in SPF mice here. So, and this is very interesting. You can see both in the column. So when this suggested that when they have the microbiota, they induce the genes, perhaps to evade the microbiota competition. And indeed, if you look at the amino acid levels in the second or in the column, you can see with the exception of 2 amino acids, all the amino acids are depleted. You see that? Because the microbiota basically is consuming them. So this is the concentration, more concentration in red, in blue, very low concentration. All these amino acids are very enriched in gene-free, but in the columns and SPF mice, they are very low because basically the microbiota is using them. So an idea will be that the microbiota using them and starve the pathogen. So the pathogen now have to make their own amino acids to survive. If you now knock out that pathway, of course, they cannot survive, so they die. So and this is actually, we noticed that this is in vitro experiments. We generated reporter strains with these genes, and we noticed that when you remove the amino acids like this medium, they induce the genes. But you put now 2% amino acid, they shut off the biosynthesis. So basically sensing the concentration outside. When they say it's very low, now they make, okay, we have to make our own. So you just make it up to survive. So that's basically what it is. Now, if this is true, you would expect that you give them high diet rich in proteins, that you will basically enhance pathogen colonization because it will be more available, right? And this would happen. So you give them, these are different concentrations of the pathogen, 10 to the 7, to infection 10 to the 6, and 10 to the 5th. You can see early on, there's some difference. This is about 3 logs difference early on, but later on they catch up and basically no difference. But you give them 10 to the 5th bacteria, so now you only can infect the mice which have been given the 50% protein diet versus the 14. So the 14 normal chow, they don't infect at all. So it makes a difference. It can make a difference. So what I'm telling you so far is that there are different levels of colonization direct mechanisms. One of them is competition for nutrients. We think that dietary amino acid competition for dietary amino acids is very important. We know for Salmonella and for Citrobacter rodentia, but we think in other pathogens also that are important. I don't say all the pathogens, but for many pathogens. This is one mechanism, and basically the pathogen now basically induce their own biosynthesis to at least some of them they can survive the infection and replicate. The other mechanisms I'm going to discuss today, perhaps more interesting one is this one, because bile acids kill Clostridium difficile. That's what we use now, fecal transplants in our hospitals, basically to treat patients with Clostridium difficile because that is a major problem after use of broad spectrum antibiotics. So some of them become resistant to antibiotics, and now effectively we treat them with fecal transplant and restore bile acids, and that kills the pathogen. So this is used in the clinic. This is probably the only example of widely used of this not of knowledge on colonization resistance to treat patients in the clinic nowadays. Is basically patients with that disease that we treat them with fecal transplants, and it's very, 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 very effective. Now, I'm going to move now, in the last, hopefully 10 minutes or so, to the other side. I've been telling you the protective mechanism, what are good for us. These microbiota really help us to fight pathogen infections. But there's a dark side, and one of these is Crohn's disease. So this is a chronic disorder of the GI tract of unknown etiology. This is really common in Spain and in European countries. We thought it's driven by a so-called immunology called Th1 T cells that in response to members of the microbiota. This have transmural inflammation. You can see an ulcer here, a very narrow ulcer. These blue dots are lymphocytes, CD4 cells. You see the ulcer here, transmural inflammation. The other thing is that this is segmental involvement. Some areas like small intestine, or this is an area in red, but the other area is basically normal. So this is very different from ulcerative colitis, in which basically go from the rectum up, but it's continuous, and it's very surface. This is transmural and segmental, okay? So we really don't know the cause of this. What we know is that many genes, more than 150 genes have been mutated in this, and people think there's some dysbiosis that perhaps could contribute to this, but we really know. Indeed, there's some alterations in the members of the microbiota in patients, but we think that most of this is secondary to the inflammation. The increase of proteobacteria, often when you have colitis, heme in the hemorrhagic bleed, many bacteria take advantage of that and they bloom. So we think some of these alterations are secondary, not the cause of the inflammation. Now, this is the challenge for the immune system. You have 100 trillions of microorganisms, many of them bacteria and viruses. The only separation between they and us is the mucous layer and a single layer epithelium. This is an incredible challenge, because they cannot tell a pathogen from a non-pathogen. The recognition of pathogen is not based on a pathogen-specific recognition. It's basically on things like LPS, which are present in every bacteria. So how do you deal with this? So there was a theory a long time ago that they think, we are tolerant, meaning that they go across and we just, well, that's not true. You inject these members to a microbiota, to a mouse or a human, and they produce cytokines, they produce antibodies. They're not tolerant, okay? Well, the way we have dealt with this is that we try to build mucosal firewalls in which we produce antimicrobial peptides, IgA, many types of antimicrobial peptides, and then the mucosal layer. By doing that, we basically limit the penetration. So basically, we separate them from us as much as we can here. Now, many of the genes which have been shown to be related to Crohn's disease or ulcerative colitis are mutations of these mucosal firewalls. So basically, many of these genes shown here. Some of them, they go across, and then neutrophils, they have genes here, which you mutate, and they're important for the killing when they go across. The few they go through, they are killed effectively. So we don't have colitis, basically. We separate them, and then some of them go through, we just kill them very effectively by macrophages and neutrophils. Many of the mutations that cause Crohn's disease are mutations that regulate these killing mechanisms. Okay, suggesting that's a problem. They go across now and they produce inflammation. Okay. Now, two members one member is important is NO2. This is sense bacteria, and they are in Paneth cells and other cells. And this is being now validated by many studies. The other one is the NAPDH oxidase. This is a part of the complex in neutrophils and macrophages that kill bacteria inside of the neutrophils by producing ROS, reactive oxygen species. And we said in the laboratory about 6 years ago, let's do the following. Let's mutate singly and doubly, so we destroy two mucosal firewalls, and then we put different microbiota to see if they now develop Crohn's disease. Because these genes are involved in human disease. If we mutate them in a mouse and we really disable two major, they should have a chance to develop Crohn's disease. Well, Roberta Caruso came to the lab from Rome, is a gastroenterologist. I just said, I'm going to take this project. Took her 5 years to complete, because she did the following. She got mice, double knockout, single knockout, and mice. And then she fostered them to recipient mothers, recipient dams, within 2 days after birth. Why? Because the same mouse was basically fostered to a member of Taconic. We have different microbiota from Jackson mice. That way we have the mutations in different types of microbiota. So we cover more. We also microbial factors, okay? So this is a lot of work, because you have to now to foster within 2 days, because then acquire the microbiota of the foster mother, not of this one. So now we have the same mutation in two different microbiotas. And sometimes we use the litter mates from the same mouse. They're brothers and sisters. And then we wash them. To our delight, one group developed Crohn's disease. What are these mice? The double knockout foster to the Taconic microbiota. 100% penetrant. Some of them develop a little later, but 100%. The same mutation when they foster to Jackson mice, no disease. This fits very well with the genetics of Crohn's disease, is that sometimes it's clear that you need the mutations, but also something else. We think that you may have the mutations, but you don't have the right combination of the microbiota, you don't have the disease. Sometimes twin brothers, there's not 100% same. Sometimes twin brothers have disease, the other one doesn't, and they have same genes. So how this happen? Well, like humans, they have skip lesions. In red, areas involved, in green, uninvolved area. Look, this is the normal Jackson Taconic mice. They don't develop colitis. You see normal epithelium in the column. But look here, the involved area. Ulcer here, transmural inflammation, a lot of inflammation. You see the so-called pathological regenerating glands here because they've been damaged here. About 20% of the second and colon are involved here. So it looks pathologically like Crohn's disease. What kind of cells is this? What we found is that the cells that increase the most are CD4 T cells, like in patients with Crohn's disease. And also early on, like in humans, there's an increase in neutrophils. These are represent early Crohn's disease. And a few cases have been studied in humans. They produce TNF alpha and interferon gamma. So this is basically the signatures of the immunologist called Th1, like patients with Crohn's disease. And this is the RNA, and this is the protein level. I'm going to give you much time because we're running out of time. So how do you now find the difference, the macro triggering this response in the presence of these two mutations, which presumably is present in taconic mice, but not in Jackson mice? So we study, we look at, so 100% of the mice develop, this is like procallic, it's a marker you do ELISA, it's a marker of inflammation, so we can do quantitatively, we can measure the colitis. And then we look actually at these mice, because before they produce inflammation, because otherwise you will not know if the changes in the microbiota are secondary to the inflammation. Okay, these mice, they develop two weeks later the disease. Well, I'm not going to give you this one member of the microbiota, this one, increased about more than 1,000-fold here. This is 1,500-fold. This is about 1,400-fold increase. Now, where's the comparison? This is the taconic wild type versus the taconic double knockout no inflame. So both no inflame, both are taconic, but this is double knockout. So clearly the genes control this. Okay. What is it? The bacteria, Mycobacterium slenderi. This is a bacterium live very close to the epithelial lining in the mucous layer. Okay, and it's a gram-negative, strictly anaerobe here. We sequenced this genome of this microbe. We found that you see in the inflamed and non-inflamed is present. This is in the feces, and lasts less in the single knockout. But in Jackson is missing here. And we found the DNA in neutrophils in the inside of the lumen and also in the laminar propria. This is the definitive experiment. So now, how you prove that this is the bacterium? Well, you put now this into Jackson, good luck this, and then we introduce by gavage, and now they develop colitis. The Jackson develop colitis now. Okay, but only in the double knockout, not in the single. Okay. Now, I don't want to tell you that, but this involves a part with the neutrophils, and we don't have time to show you more of this. But one thing that was very curious, I told you at the beginning, these skip lesions. Why is that? People are mysterious. All the gastroenterologists always, it's a main feature. That's what they separate often Crohn from ulcerative colitis, but they don't know why this. Well, we reason that perhaps this represents local penetration area in some areas. So this is the experiment. 2 mice here. This is the inflamed area. We took it out with a laser capture mechanism. So this is after the laser capture. See, the tissue is gone. Go to a tube, test tube. This one is neighboring, no inflamed. Took it out. What represent this contains the DNA from the bacterium. So this is the control here, okay? So it appears, at least in the mouse model, that the skip lesions are area which is no penetration. And the areas of penetration is the inflamed area. It makes a lot of sense. But nobody has been able to prove that. And we think that this is probably what happened in humans with Crohn's disease. The problem with Crohn's disease is that it could be a number of bacteria. This model suggests that one bacteria is sufficient to give you the disease. And it could be very rare, and that's why nobody's been able to find it. And there could be more than one, depending on the patient, depending on the mutation. So that's been the challenge that we've been able to find, because that would be, in order to have an effective treatment, you will have to find these organisms in patients with Crohn's disease. So I think this is the last slide of this. So in wild-type bacteria, in wild-type mouse, like in humans, there will be some bacteria living very near the epithelium. In fact, this bacteria elicit IgG in all the mice because probably it's passed across, but because you have NOT2 and you have neutrophils killing, then you don't have any inflammation, but it's all control. But if you now have mutation that destroy 2 main mechanisms that control members of the microbiota, they can have ability to penetrate, like this one. Now they increase the number, and then some of them penetrate and produce something that we call, at least in the mouse, Crohn-like disease. So that's basically what I want to tell you. And the other thing is we have now found ways to change this with diet. With diet, we can control this disease by understanding the metabolic pathways of this organism. We can treat effectively with diet and prevent the disease. And you have more, I can tell you that. Anyway, so I told you already everybody who worked on this. This is another big picture. This is a few years ago. And some of our members help us José Luis Puente from Universidad Autónoma de Mexico, and Kathy Negler from University of Chicago. And I would say Nagohiro Nohara, I mentioned Erin Martens, that I mentioned. All the others are being mentioned in my talk during the presentation. Thank you very much. So for the questions, I like the students to ask me the questions first. You can do this in Castellano, there's no problem, or in English, or probably in Italian perhaps. Catalan, sorry, I don't understand Catalan, but I will try if you ask me. Do my effort to do it, okay? Questions for the students? Come on, you have some questions. You know, when I'm in Sevilla, I invite to the restaurant to the student ask me questions. That sometimes works. No, students, come on, don't be shy, come on. There's no stupid questions, I always say that. No, they are very shy, okay? Oh, good. One question, make my day, good.
How many bacteria do you think that is killing the stomach from the oral to gut route?
So the question is how many bacteria are in the stomach?
Yeah.
So the stomach, as you know, because the acidity and the environment, the density of bacteria in the stomach is less. So about 1,000 more, less than in the distal intestine. That's the question, how many bacteria? And also the other thing is that the microbiota of the stomach is very different from the small intestine, from the large intestine or the small intestine, because the nutrients are very different, and they are different. Is that the question you want to ask me?
Yeah, and how the bacteria arrives to the gut if they cross from the stomach and the acid pH.
Yeah, this is very, very interesting. So when we people work in the oral infections, what we do become bicarbonate. You know, the old-fashioned bicarbonate with people here in Spain when I was growing up, they bicarbonate, you know, that's what you do. So that affects a little bit. So I think because we give in such a high dose, like 10 to the 8, 10 to the 9, even if we kill 100-fold, they still have 10 to the 6. So it's enough threshold to get that infection, considered with Citrobacter rodention. So we can, to increase a little bit, we can give them bicarbonate, and we increase between 5 and 10-fold. So we make them increase the dose by doing that. So some people, they give them bicarbonate to basically to increase the pH, so some of them survive better. But because we give them so many bacteria, even this is very, very quickly, by 30 minutes already go through the stomach, very quickly. You can detect them already, some go to the small intestine. So they can go very quickly. So some of them die, but we think probably between 5 and 10-fold only, okay, of the bacteria we put in. They die in the stomach because the acidity and other factors. Yeah. But we do this. Often people do, they give bicarbonate before the infection to increase the number of the bacteria that can reach the large intestine. Yeah.
Thank you.
There's a question in the back.
Hi, I was just wondering, have you seen any differences, not only with disease, but also like in health, maybe in different stages of life, like in when you're old, and like how the microbiota changes the interaction with the host?
Yeah, this is a very good question. Well, we know, I don't work in this particular, but what I read is that we know that microbiota is very resilient. There's no much change. Here, everybody here have different microbiota. We can tell you do heavy Illumina sequencing, who is which here. I mean, you eat a diet which will change the microbiota more quickly, right? Within 24 hours or less, you can change. But then you are vegetarian, you start eating meat, you change the microbiota very quickly, you go back to vegetarian, and then you go back to normal, okay? So, there have been changes reported in the literature of the microbiota of age. Let's say centenarians have different microbiota. But the problem with that is that centenarians have a different diet than they when 40 years old. Basically, most of them don't have teeth anymore. And often they're eating very soft food. Of course, you change the food, of course they're going to have a different microbiota. But I don't think it's very good evidence that aging itself changes the microbiota just because they age. It's very difficult because the diet is such a strong factor in composition of the microbiota that you change the diet, of course, you're going to change. It happen to be linked to different diets of old people. So I think, although there's some evidence, that there are some changes, but it's relatively minor compared with the difference variation between individual to individual in this particular age. But yeah, it's very complicated. It's very complicated to study in very old people because they change the microbiota and the lifestyle, which can impact on the microbiota. So is that age or is actually the different lifestyles now with different food? It's a very tough thing to differentiate. Now I guess you got now people 30 and older. The centenarians are here in the front. They survived already.
Hi, I have a question. Maybe you already explained this here.
Yeah.
But I lost it. In Crohn's disease, in the affected regions in the mucosa, I didn't understand if there are somatic mutations of the host in humans or germline mutations that have been observed in these regions.
There are germline mutations.
Germline.
Yeah.
Okay.
So in contrast to cancer, that's somatic mutation driven by the tumor. In some cases of somatic mutations, some diseases, these are germline mutations. So you inherit it from your parents. In the case of NOD2, to give you about 20-fold to 40-fold increased stability, you need to have homozygous. It means that two alleles that are mutant from your mother and your father. So you basically become like a hyperresponsive or a knockout. Or a compound heterozygous, meaning your parents have different mutations. In this room here, 20 or 30% of you have the NOD2 mutations, but you don't have to worry. So there will be 20 people here who have mutations or more. But because you're only heterozygous. So it's not much in fact, okay? Smoking gives you more stability than that. But to be a compound, you need about 1 in 5,000 individuals to find one. Even that, the penetrance is only 5%. Because you presumably like the microbiota, like the Jackson mice. That they have the mutations, but they don't have the microbiota, presumably it could be something else. They don't have the disease. So it's a very low penetrance. But this is a classical genetic disease which are genetically complex. This is not like cystic fibrosis, that you get the mutation and that doesn't matter what you do. You get cystic fibrosis. Nothing you can do about it. You will get eventually, maybe it depends on 2 or 3 years, but you're going to get cystic fibrosis. You have the mutation. In Crohn's disease, it's not. It is genetic important, but also environmental factors. Both have to be together to develop the disease. Okay? Complex genetic diseases.
Thank you.
Thank you for the talk. So you mentioned in your TN6 study that your bacteria needed amino acid production to colonize the gut. So I was wondering, did you check if those genes were present in other common gut pathogens?
If this mechanism?
If this amino acid production is also present in other bacteria that are commonly found?
Yeah. Well, this is known because you have how many genomes you have in the deadline. You can analyze many of them amino acid biosynthesis, many bacteria in the gut. So you can find out because all these genomes are already online, so you can check. They are basically typically it's an operon which are different enzymes to make an amino acid from a single precursor, so you can make amino acids. So not all the bacteria, but many bacteria, and also the percent have operons for the biosynthesis of amino acids. Certainly Salmonella. There are many bacteria, but yeah. I don't know the numbers, and probably someone with studied the genomes of bacteria on a daily basis to tell you, but this is, many bacteria can make their own amino acids, yeah.
Thanks.
Yeah. Here. I don't have the microphone. And again, you can ask the question in Castellano, okay? Maybe in Catalan also, because maybe I can practice a little bit. Maybe I can.
Well, thank you very much for the nice talk. Talking about the dark side of the microbiota, could you comment a little bit on the syndrome of bacterial overgrowth and if it can be prevented?
Are you referring to when they have the loop, in what situation? Because there are several pathologies which are associated to bacterial overgrowth, right? So
the one that colonizes the small intestine by microbiota from the large intestine?
Oh, yeah, because there's some surgeries that, yeah, I know really an expert, I really have not studied that particular, I know that this is a very important question, and there are many pathologies that they're associated with that. And I've been learning this a lot because I'm a senior editor of gastroenterology, believe it or not. I'm not a gastroenterologist, but if you send a paper to gastroenterology, it goes through me, okay? Particularly it's a basic paper, okay? So, but yeah, so I live in the, and we know since our medical school, there is a pathologist with diarrhea and so forth, and there's some treatments. You are a gastroenterologist, you probably much know more, more than me. But there's some, I saw some papers in which people try to use certain strategies to diminish the colonization by that. But I'm not sure how effective that is. But there's some literature, but I don't work in this particular topic. But this is clinically potentially it's a very important problem in some patients. No question about that. And are you a gastroenterologist?
No.
Okay.
No, not at all.
Yeah. So I know the gastroenterologists treat these patients because these are things in the clinic, and this calls out sometimes for special surgeries and things like that. Yeah. Basically, the environment of the small intestine changes because they connected with the colon, for example, and now the pH is different. So that makes major artificial alterations, but clinically very important for the patient. But there are some strategies to minimize that and treat these patients, which really affect the quality of life. No question about that. Yeah.
I have two questions for you.
Mm-hmm.
The first one is, it's like if you say that both the genome and the microbiota in the intestines, it's what you need to trigger IBD. Why not treating people with Crohn's disease with antibiotics makes them get rid of the disease,
okay.
Right?
Yeah.
And the second one is, what makes these bacteria so special to induce this or trigger this disease?
Wow, those are wonderful questions, which sometimes I ask myself. So the first one, yeah, there's some studies, typically with single antibiotics. If you treat with single antibiotics, actually, you're probably going to diminish bacteria. In fact, some patients I will predict they will get worse because you killed a competitor, they go up. So I don't think some patients have been claimed that have improved antibiotics, but you cannot sustain that because they become resistant. But if you, for example, some model we have in the lab, if you give even one single, it can worse because the bacteria now, when you kill the competitor, it gets worse. So I think there's been never a consensus because the results in patients were so mixed. Some people get better, other people get worse. So they said, well, we cannot use this, it's not effective. And it makes a lot of sense, the way you are. So what I propose to do, is do the following, is to first identify the microbes that cause the disease. And that is very complicated. So we have to do a different kind of approaches, probably like we did in mice. Before they get patients that will develop Crohn's disease, but before you got the flares, you can get samples sequentially. And then to see this. And then when you isolate this organism, which will take some time, then you can find metabolic pathways which are required for survival. And you can make a specific antibiotic. We have done that with Mycobacterium. Okay, we know a pathway with nitrite metabolism, which is very important. And we can make a drug that can actually kill it without affecting other bacteria. The advantage of that is that it's very specific, and also these enzymes may not be present in the host, so it's no side effects. But to do that I think will be the future, 20, 30, 40 years. First, we need to identify the organisms, and there will be probably several. And then we can make a specific antibiotic to control the patient. Then we'll have to do trials. Anyway, so that will be like that. Now, what is the special about, that's a very important question. So far, with a few cases unknown, they live very close to the epithelium. It makes sense. If you live in the lumen, it's not in the health. You wait, that you're going to develop Crohn's disease because you have to penetrate. I mean, they cannot send something for the T cells to activate. So they have to penetrate. So I would say this one also have some virulence factors, like a type 6 secretion system. So I think there will be bacteria which are microbiota. That's why they don't go to the liver. They produce inflammation in the intestine because that's where they live. But they have to live very near the epithelial surface, so they have a chance to penetrate. And this one lives in the mucus, so it's very close. And then they have some virulence factor. They survive, perhaps neutrophil killing a little bit, whatever, to cause the inflammation. That's what I think. But we don't have direct proof. The only proof is this one. Yes, they have some virulence and live in the intestine. I think living near near the mucus, living near the epithelium, it makes a lot of sense. Because if it lives in the lumen, far away from the epithelial surface, there's no way they can cause inflammation. It's not possible. So I think that they have to have a lifestyle. They live very near the epithelial surface. This particular bacterium, the reason why they live in the epithelium, because they cannot digest the mucus, but requires mucus products from other bacteria, which are is glossing together with other bacteria. They can now digest, hydrolyze this mucus, and then products are used by this bacteria. So they need to be together. We have work on that. I don't have time to show you, but we have a lot of work done on that. It's unpublished, yeah.
Have you tried using other bacteria that lives close to the epithelium or not to see?
Yeah, well, I mean, so many bacteria have been tested. Many they live in the mucous layer. In this particular model, that's the only one that causes the disease. We tested, not all, but we tested many, and they just doesn't produce the disease. So I think there's something unique. But some of that makes sense, but we don't really know for sure. We don't have direct proof, basically. We cannot make a mutant that they can abolish this. The only to prove this, in this bacterium, will make a mutation, they cannot go to the mucus. And then to show there's not, with good colonization, there's not. We have a correlation. With a particular diet, we can move into the lumen, okay? It's not Crohn's disease. This goes away, okay? That's the only, but we cannot make a mutation because we cannot genetically modify this bacterium yet. Yeah. Yeah.
Continuing with this, the same topic, what kind of sampling would it be needed for making this identification of the microbes? Yeah,
I think my idea, they have this idea, and sometimes I write a review and I say that to see some gastroenterologists sort of take the idea. I'm a pathologist, I was a gastroenterologist, perhaps will done the study. Basically, what I think is that you have to have a cohort in which genetically identical. So you will use probably patients with not two mutations, is most common. And then I will basically select sequentially, get samples. Ideally, it will be biopsies, but maybe the fecal will be sufficient. But then, and then capture them, patients that will develop a flare. So before you develop the flare, you get the sample. And then to see if now the increase within the flare. So you probably sequence by Illumina sequencing, and you do this in a cohort that is likely to develop a flare. And then you do this in many patients which are genetically similar, okay? And then I think we'll have a chance. The only thing I want to tell you is that I think that the bacteria that cause ileitis and rectal disease is different in our model. We have now a model, they produce ileitis, like horrible ileitis. It's never seen in the mouse. We've seen it. The bacteria is different from Pseudomonas sandeli. That lives in the small intestine. So I think this may be applied to humans, that you have rectitis, and the bacteria in the ileum is different. I mean, it is not proven yet, but that's my prediction, okay? But that's the way I will design the study. If I have a patient group, and follow before they get colitis, and they will get sequentially. And then you're lucky some of them develop colitis, and then you examine massive analysis to see what's going up and down. And then you find correlation with more patients to see if the flares correlate with increase. I think at the end, gastroenterology in Crohn's disease, I will see like the rheumatologists in 100 years or 50 years. You come to the patient, you have a patient with Crohn's disease, they can give you here, you get a fecal sample. You have now sequencing, total sequencing. I tell you which one of the 20 ones that cause Crohn's disease are there. Okay, you have pathogen number 5. And then we're going to follow that. So that's the way the gastrology will practice in 50 years. We already have the list, hopefully, and they say, which one do you have? Okay, you have that, okay, then the treatment is this, okay? Because we know there's a drug to kill that one, this antibiotic to kill that one. And then we will follow you. That will be the practice on IBD, okay? So this is like in the future, okay? I don't think I'm going to be alive when this happens, but hopefully.
Hello. Thank you very much for the talk. I was wondering, since you mentioned that there is this contribution from the host, like regarding mutations, on, I'm guessing, like there's multiple genes, there's no few of them, but the other contribution is microbiota, and you mentioned that it changed dramatically when you change diet. So there's any strategy? I mean, if you know, for example, you have the mutations or you have family members that have this kind of disease, changing diet, it's recommended to prevent the onset of the disease, or it can change the course of the disease when it's already started? Thank you.
Yeah, that's a very good point, very good question. The problem is we don't know which is the microbiota in patients with Crohn's disease. So the diet can diminish. We know that can change the microbiota a lot, but you don't know. Maybe the diet you take increase the pathobion. So, because we don't know. If we know which one is causing, you can follow it and say, okay, that diet increases that, don't eat that. Don't eat chocolate because that likes that. So don't be, by the way, yeah, when I eat chocolate, I get worse. Okay, makes sense now, right? So yeah, and some people have claimed that they avoid certain foods, for example, to, fiber uptake is sometimes patients get worse. Patients with Crohn's disease. But we don't know. This is a little bit anecdotal information. I think once we know which are the bacteria, then we can do these sort of studies in a logical way. But yeah, you would predict that certain foods you will get better. And I told you there's only if one food will worse in Crohn's disease in pediatric Crohn's, which is a fiber-free alimentary exclusion diet. And that's used in Europe, not in the United States because the insurance company doesn't cover that. It's a 7 or 8 week course. And it's a good at corticosteroids in children with Crohn's disease to induce remission. What we found, we give this diet to the same we give to the patients, to this model, the Crohn's disease goes away. Why? The postperiod goes to the lumen.
Thank you. Very nice talk. It was pleasure. I have a very specific question about the Crohn. You maybe have mentioned it, but in your double knockout model, if you do a fecal transplant from Crohn's disease patients, do they develop the symptoms?
We haven't done it in this model. What we know from the literature is that in Crohn's disease, fecal transplants don't work very well. In ulcerative colitis, some patients benefit. And you have to do this weekly, something like that, or more than once. So in contrast to Clostridium difficile, which is just do one transplant. So in some patients with ulcerative colitis benefit from fecal transplantation. They get better. Some patients, and requires, because the microbiota needs to be changed, right? So in Clostridium difficile, the microbiota is totally, the diversity is greatly reduced. So you're giving a fecal transplant, you restore the diversity. But in patients with Crohn's disease and ulcerative colitis, the diversity is diminished, but it's not as much. You know, they're just different populations, probably secondary to the inflammation. So we have not treated our mice with fecal transplant. So I can now tell you that. What I tell you is the data in the clinic. There are many studies already published in patients with that. Yeah.
I was wondering, like, to try to identify the bacteria that trigger that, right? Like doing a taconic microbiota, a Jackson microbiota, but with patients that have Crohn's disease that probably have those bacteria that trigger the disease.
Exactly. This is what Roberta is doing. And she has very interesting data. When I tell you she does, she's not hearing me now. So basically, what we have gene-free double knockout, and we're putting the microbiota from patients, and they develop Crohn's. Yeah, and we just have few cases, but some of the control microbiota or the wild-type mice doesn't. So we're looking for that. So yes, we have actually, some of them produce very bad Crohn's disease with microbiota from. These are pediatric patients, by the way. Pediatric children with Crohn's disease. We put in a double knockout. We put this, and they develop colitis. Yes, but we don't yet know which one it is. But that's yes. So you're totally right. That makes a lot of sense. So she's doing now that her project, and she's going to continue that on her own. So that will be perhaps one way to identify the microbes. Yeah. Totally agree. Very good question. Yeah. Thank you. Wow, this has been, I was told when I came here, said, there are going to be no questions here. They're kind of shy. I think you're telling me that, right? You tell me, yeah. The students don't ask any questions. They are kind of shy in Valencia. Good. Oh, another question.
So, Gabriel, thank you for the talk. So, considering that us as a healthy individuals, right? So we have a continuously colonization resistance. So what do you think about taking in a regular manner probiotics? And does that going to have any effect on our immune system, or actually not, because we have colonization resistance and we suppress their colonization?
Yeah, well, I don't want to take I think generally speaking, all the studies published, with very few exceptions, the probiotics introduced singly, particularly in the absence of previous treatment with antibiotics, they don't last very long. In fact, all the yogurt company have been trying for years and years to publish one single paper showing any beneficial in a controlled way. They don't have any proof, otherwise it will be all over the place. I mean, they don't colonize. And if even, you remember these probiotics were basically introduced in the early 1900s by Eli Mexensoff. He basically went to Bulgaria and he found Lactobacillus bulgaricus. This is one of the main the yogurt industry uses. This is basically taken from peasants in Bulgaria that basically were over 90 years old. They were centenarians, some of them. And they were basically using milk from cows, and they were basically fermenting it. So he basically, Eli, all his life, when he became director of the Pasteur, he was using Bulgaricus. I mean, basically in his house, he would basically will ferment his milk, and they will use this every day. He died in the 60s of a heart attack. Okay, fine, didn't work. But when he did many studies, and later on it was shown that they never could find in animals Bulgaricus, never. So they were having some effects, but actually the bacteria is not even there. So this probably was caused by variation, some sort of random variation, and they were claiming some experiments some benefit, but actually the bacteria was not even detected anymore. So there's many publications from the Israeli group. Perhaps they have a better study, and many others. So I think you will have to have these in a daily basis, or maybe twice a day. And then the question is, what is unique about this bacteria? We have already hundreds of trillions of bacteria. How one bacteria is going to be better than the one we have there? It doesn't make any sense to me. I mean, if you modify genetically the bacteria, do something that we don't have, perhaps will make a sense. But why are you going to put a bacteria which, as far as I know, many other bacteria in there, they have the same genomes and the same pathway. Why this bacteria molecularly is going to do something different than the one we have here? It doesn't make any sense to me. Yeah. That's what I say. And then on top of that, they don't last very long. You can take, you can go to the supermarket, the probes, you can inject yourself daily yogurt and breakfast and measure 2 days later. They're not there anymore. They're gone. I think this is just fallacy. I would say that maybe eventually someone, maybe engineering something, but I think the way prebiotics make more sense. Prebiotics, basically, you use the same bacteria to feed them, to bloom them. Yeah, that works very well. So if you know which is the group of bacteria that is beneficial, okay, give them the food they need. They will protect you because they will go up by 100-fold or 10-fold. So prebiotics, I think, is the way it should work because that works very well. And also it's transient, right? So you remove it, it goes away. So that way you don't have any side effects. You can control that. Prebiotics work very well. This has been shown very well. So, but the question is, which are the bacteria you want to increase the number? Make sure you don't make a mistake, right? So once you know which are the good ones, you can give them, you can feed them, and you don't have to inject anything. That is more natural, right? That having something endogenous, putting in there, just take care of your own bacteria. Right? Just select the ones you have there and give them. They protect you for something you increase. That makes more sense to me, and it's very, very effective. You can increase the numbers. They're already there. They already have their own interactions with the bacteria there. They just go up. If more food, they go up. If they do something, I think that makes more sense to me. But a single bacteria, I mean, a big consortia like fecal transplant, yes, works well in people having already have very low diversity, or then yes, because you put now a whole complex microbiota into, and that will take in. But a single bacteria or 2 or 3 bacteria by itself, there's no chance to compete with already well-established microbiota there. It's just not possible. That's my view, okay? And that's supported by the data, okay? You know, but someone had to publish something different to contradict me. Okay.
I guess we finished the talk, no? I
Yeah,
don't know if anybody has any question.
We're going to increase the microbiota, paella eating microbiota.
Exactly.
I think there's some, I think the certain foods in the paella valenciana, they love that. I'm sure we're going to increase, by 5 o'clock, we're going to have certain bacteria which are going to bloom with the paella.
The garrafón bacteria
is very good for
it's going to increase. Well, thank you very much, everybody. And thank you, Gabriel.